Hello. Today we'll be talking about molecular biotechnology. We'll be talking about what it is, what are recombinant proteins, which are very important. We love those. They're used all over in the industry. It's one of like the most valuable industries out there, actually. We'll talk about what are the differences between transient and integrating expression vectors. Integrating expression vectors are generally preferred when it comes to large scale, like in your bioreactors or whatever, and we'll see why later. How do we make these vectors in the first place? The expression hosts that we need and that we can use. We'll talk about four main ones today. They're all pretty cool. They all have their pros and cons. And how do we actually express these human therapeutic proteins that are so valuable? So, what is molecular biotechnology? It is just simply applying a design genetic change onto a host organism, and then we get a useful product out of that is produced that we can purify with the downstream biotechnology, right? So insulin, we get that from cultures of E. coli. This other drug, Herceptin, we can get that from cultures, and then biodiesel. We actually do engineer E. coli to make more precursors for biodiesel or the molecules that make up biodiesel, right? So that is still molecular biotechnology, even though we're not getting a drug out of it. Um, so we'll go over expression vectors real quick. It is a DNA plasmid that could be transformed into a host cell, right, which contains your foreign DNA that you want to be expressed. This is called heterologous expression because we're expressing a foreign piece of DNA that isn't originally meant to be expressed in the host. Um, and what a plasmid is like an external form of DNA that is normally found in bacteria. And why they do this is because the plasmid stores excess genes, and if it was stored in the actual genome, it would slow down replication, right? So the plasmid is only really used in like your stationary phase. So stuff like secondary metabolites would be expressed through the plasmid, as they're not necessary for growth. They're just an addition that can help them gain resources during the stationary phase. So, for example, penicillium would have the penicillin gene on its plasmid. I think, I hope. Maybe I'm making that up. I don't know. Okay, and here is your plasmid. Here's what they look like. They have a selectable marker, antibiotic resistance gene. Actually, yeah, key features needed. Selectable marker, correct stabilization sequence, so poly and capping and whatnot. So you just can't have a bare sequence. It just won't. It will be broken down as when it goes to mRNA. And ORI-C, right? If it's like non-integrating, so it can replicate independently of the chromosome. Um, and a multiple cloning site. This is for when you use restriction enzyme cloning, which is kind of like a dated method. There's another method that we'll go over soon um, called that uses homologous recombination events. So what is the difference between integrating and transient expression vectors? In transient expression vectors, the plasmid is separate from the host cell genome, right? So they don't replicate together. And this is good for small scale systems, um, but it's not really scalable because you have this thing that occurs that's called vector loss or plasmid loss, and that is when the host's um, genome replicates faster than the plasmid. The cell separates, and only one cell has that plasmid in it, and this other cell is plasmid free. You keep having that in a large bioreactor, you're going to get very poor yields. But to compensate for this, they use high copy number plasmids, and this is just like having a fast replication time. And for reference, this is like what defines a high copy number plasmid. Um, yeah, just how much DNA it is. And then an integrating vector actually becomes part of the host genome. This is mandatory for industrial upscaling. You get a low gene dosage effect, but this is remedied by using really strong promoters. You can either use ubiquitous ones, um, or others, depending if the protein expressed is actually toxic for the cell. If it was, you wouldn't use a ubiquitous expression, right? You'd wait to switch on expression whilst it's in that log phase, whilst it's in exponentially growing. Some systems that I use are BACMID and T7. I'll explain BACMID a little bit, but I won't get into T7. But I'll leave a link below that will tell you all about it. It's pretty fun, pretty cool. And this is like a cool little diagram for stable transfection, transient transfection, long-term protein expression. Long-term, we love that. How do we clone our gene of interest into our vectors? So, as earlier said, you can use a restriction enzyme, which is where a donor plasmid is created, where the gene of interest is flanked by specific restriction recognition sites. So both the donor plasmid and the expression vector are incubated with these same restriction enzymes, so they get cut the exact same. The gene of interest is then isolated and introduced into the vector with the addition of ligase. So ligase, gene of interest going here, ligase shuts the gap. And then phosphatases prevent reannealing, that initial reannealing. Like you just imagine you just cut it, right? You cut your donor plasmid and then it reanneals. There was no point of cutting it. You just wasted your time. Now we have the more, would I say sophisticated? I would say I like this method more. It's called gateway recombination cloning, where truncated inserts are avoided and the gene of interest is flanked by ATL sequences, which are identical to the sequences in the expression vector. So you have your gene of interest, you have the sequence that you're trying to knock out. These sequences right here are the same and they cause a recombination event. So your gene of interest is inserted into the genome. And so both of these plasmids are incubated with enzymes necessary for the recombination events to occur. A clonase mix, and this is shown here, little boop, boop, boop. It's like Legos, basically, DNA. There we go, recombinant plasmid, wow. Now, how do we get this plasmid into our expression host of choice? We can use a gene gun, which is particle bombardment, where you literally like shoot the DNA into the cells. You can integrate it. Wait, yeah. Where you identify an endogenous gene in your expression hosts, use strong promoters, and flank your gene of interest with a homologous sequence that flank the host gene. This is basically how you get your gene into the host genome. You can have baculovirus transformation, where the BACMID is a shuttle vector that allows viral DNA to be inserted into the host genome, right? Because they express this thing called polyhedra, which is needed to protect the viruses that are expanding in the host that are being made. So instead of this polyhedra gene, which is on a strong promoter, you just insert your gene of interest into that via recombination. And physical methods, you can just like inject your expression vector into there. You can electroporate it, or you can use silicon carbide whiskers, which poke little holes into it as well. So this is all the ways you get your vector into your expression host. Well, there's more, but you know what I'm saying. Now we go over the different types of expression hosts. And first we have E. coli, the LeBron James of molecular biotechnology. This guy's goated. It has a very tractable genome, so very easily manipulatable. We've sequenced E. coli so many times. There are literally websites out there that tell you what gene expresses what protein. Low costs, but it has no post-translational modification capabilities, so it doesn't have the machinery even to do those, which we'll see is important later on. We have yeast, Saccharomyces cerevisiae. This is a simple eukaryote. Again, very used in science. This is Michael Jordan of biotechnology. These guys are running for first place, basically. Yeah, again, tractable genome, fast generation times. And there's some post-translational modification, but you have insect lines like the full armyworm, and these can be expressed using the baculovirus, right? Uh, the only thing with this, you can't do continuous expression because the virus kills the cells over time, and it's a little bit more human-like. But the most human-like are Chinese hamster ovary cells. I don't know why they chose to call them Chinese hamster ovary cells, but I guess. Um, they have human glycosylation patterns, which I'll go over with you in the next slide, and they're used for therapeutic protein expression, but they're very expensive. So these you normally use an integrating expression host because you don't want to lose your culture and then redo it. It takes a lot of effort. So what is glycosylation, which is one of the most important post-translational modifications? So you have your like acetylation, phosphorylation, glycosylation. This is where glycans attach to your protein, and these are a type of macromolecule to aid with stability and interactions with your cellular environment. So if you don't have the correct glycans, or too much of them, or not enough of them, your protein's function will be hindered, right? And they're made of sugar residues, essentially, typically. So if it is an N-linked glycan, it is bound to asparagine, and if it's an O-linked glycan, it is bound to the amino acid serine or threonine. Pardon. So there are different types of residues which are very important. Mannose is a sugar that is N-linked typically to asparagine, right? And found in non-mammalian glycans normally. Like high mannose glycans are not good. We do not want those. And sialic acids are a terminal sugar which are only found in mammalian cells. So sialic acid residues are very important when it comes to glycosylation. And here we see some common glycans, and you see high mannose levels right here. So that's not good. We don't want those for human therapeutic proteins. And here again, yeast, high mannose, not enough NEU5AC, which is basically sialic acid, right? So here we see humans, they express those at the end. Those are very important for protein function. So which host are we actually going to choose for our therapeutic proteins? Well, this is based on what post-translational modifications they are able to do. E. coli can't really do anything, right? And it also has the annoying thing where it has pyrogen proteins that are added onto your express protein, which is cause an immune response. We don't want that. And the E. coli chaperones are not suitable for eukaryotic proteins. So the chaperones just help folding of proteins. If they're incorrectly folded, the E. coli chaperones can't help your therapeutic protein. Yeast, this has a little bit more, it can actually glycosylate, but their glycans are high in mannose, which we don't want. This is very bad. And hypoglycosylation can occur when too many glycans are attached to a protein. And here is a little article that I will link below as well that goes into that a little bit more. And then we have Chinese hamster ovary cells. These are the most human-like in glycosylation. They don't have hyperglycosylation and no contaminant proteins are attached. So it's easier to purify compared to yeast. And so that is why most people choose CHO cells as their expression host. And here we have an example of many therapeutic proteins that are expressed, right? We have Pompe disease, and you see this year of approval. It goes all the way back to 1987 with Genentech, the original molecular biotech company. So yeah, it's very widely used. You can optimize other expression hosts, so you can remove thyrodoxine slash glutarodoxine genes, so that disulfide bonds can form, right? Because the E. coli cytoplasm is not good for a disulfide bond formation, which is needed in some proteins. And here we have basically the storyline of how you get your cells, right? You transfect them, select for them, blah blah blah, screen, and yeah, and then you start purifying your protein. And that's basically it. That's basically all we have for molecular biotechnology for recombinant proteins. Hopefully this was useful. Maybe it wasn't. Uh, Apologies for any stutters as well. Okay, bye.